Welcome to the deep dive. Today, we are taking an exhaustive look inside, well, probably the most fundamental chemical factory on Earth, photosynthesis.
That's right.
We're going far beyond that simple equation you might remember from high school biology, really digging into the mechanisms, the structures, and the brilliant evolutionary adaptations that let plants capture sunlight and feed, well, virtually the entire planet.
Our mission today is to trace the journey of a single photon of light and a molecule of carbon dioxide. We want to detail exactly how life converts that raw energy into complex chemical fuel.
So we'll be looking at the key structures, the two main stages.
Exactly, and the clever metabolic compromises that plants have had to make to survive in some of the harshest environments.
Okay, let's unpack this with some scale. Photosynthesis is the ultimate energy conversion. Light energy into chemical energy stored in organic molecules. And this single process, it really defines the two major nutritional classes of life, starting with the plants themselves.
Right. We start with the autotrophs, these self-feeders. These are organisms like plants, algae, and some prokaryotes that produce their own organic molecules from really simple inorganic things like CO2 and water.
They're the producers.
They are the essential producers of the biosphere. They support pretty much all other life.
And then there's the rest of us, the heterotrophs or other feeders.
Yeah.
We are the consumers, the decomposers, and everyone else who relies directly or indirectly on the compounds made by those autotrophs. And we rely heavily on the most crucial byproduct of all of this, the oxygen we breathe.
Absolutely.
So before we dive into the chemical reactions themselves, let's set the scene. Where is this remarkable process housed in a plant? Structurally, where is this chemical factory located?
Okay, so in plants, the major sites are the chloroplasts. You find them mainly concentrated in the internal tissue of the leaf, which is called the mesophyll.
And what does that look like?
To visualize it, you can imagine a kind of fluid-filled oval container. It's enclosed by two membranes, and the dense fluid on the inside, that's called the stroma.
But the real action, as I understand it, happens in the membrane suspended within that stroma.
Exactly. Suspended right in there is a third, very complex membrane system. It's made up of these flattened, interconnected sacs called thylakoids.
Thylakoids.
And these thylakoids often stack up into columns, which we call grana. Critically, the green pigment, chlorophyll, which is what actually catches the light, is embedded right there in the thylakoid membranes.
Right. Now, when we look at the overall simplified formula for photosynthesis, carbon dioxide plus water plus light energy yields sugar plus oxygen, it kind of hides this massive, complex redox reaction.
It really does.
And the key question that early scientists faced was, where does the oxygen actually come from? Is it from splitting the CO2 or splitting the H2O?
And early on, scientists, understandably, they thought CO2 was split. It just seems logical. But in the 1930s, a researcher named C.B. Van Niel hypothesized, based on his work with sulfur bacteria, that plants actually split water, H2O, to get the electrons they need. And that releases O2 as a byproduct.
So how did they prove that? I mean, what was the definitive experiment?
It was confirmed by these really elegant experiments using a heavy isotope, oxygen-18, so O18.
They could track it.
They could track the label. They found that when they used O18 labeled water, the O18 showed up in the released oxygen gas. But when they used O18 labeled carbon dioxide, the label only appeared in the sugar and the other water that's produced, never in the oxygen gas.
Wow. So the water splitting hypothesis was confirmed. This means photosynthesis is running a massive redox process, but effectively in reverse compared to cellular respiration.
That's a really powerful way to put it. Water is oxidized, it loses electrons and hydrogen ions, and carbon dioxide is reduced, so it gains electrons and hydrogen to form the sugar.
And that reversal of electron flow needs a huge input of energy.
A massive input of solar energy. And this whole operation is divided into two cooperative stages. Right. First, the light reactions, and then the Calvin cycle.
Okay, so the light reactions are step one, the photo part. This is all happening in the thylakoid membranes. This is where the plant converts solar energy into two specific forms of chemical currency, ATP and NADPH.
To really get how a plant catches light, we first need to remember that light is electromagnetic energy. It's made of these discrete packets called photons.
Right.
And a photon's energy is inversely related to its wavelength. Plants are really only interested in visible light, that 380 to 740 nanometer range.
Why only that range?
Because only that specific energy level can excite electrons without just, completely destroying the cellular components.
And that's the job of the pigments. We see green because chlorophyll is aggressively absorbing the violet, blue, and the red parts of the spectrum, but it just reflects green light right back at us.
Precisely. Chlorophyll A is the main workhorse here. But plants have evolved these clever accessory pigments, like chlorophyll B and carotenoids.
They're like a bigger antenna array.
That's a great way to think of it. They catch wavelengths that chlorophyll misses, broadening the plant's usable light spectrum and then funneling that energy inward.
I always found the carotenoids fascinating because they also act as the plant's sunscreen, right?
That's their crucial secondary role, photoprotection. They absorb and dissipate excessive light energy that could create dangerous, highly reactive forms of oxygen and otherwise damage the whole chlorophyll machine. They were literally preventing solar flare-ups inside the leaf.
So, when a chlorophyll molecule absorbs a photon, one of its electrons gets boosted from a stable ground state to a highly unstable excited state. But inside the chloroplast, that excited energy doesn't just glow away. It's immediately put to work by these things called photosystems.
Yes. A photosystem is this organized collection of pigment molecules surrounding a central reaction center complex. Once the energy gets funneled to a special pair of chlorophyll molecules in that center, they don't relax. They instantly pass that energized electron to a primary electron acceptor.
And that's the first step.
That is the very first redox reaction of the whole light process.
And we have two of these photosystems working together in sequence. So walk us through that sequence that creates the energy.
We call it linear electron flow. It starts at photosystem 2, or PS2, which has the P680 chlorophyll pair. Light excites an electron in P680, sends it to the primary acceptor, and that leaves this massive electron hole.
A hole that needs to be filled.
And it needs to be filled immediately. P680 becomes the strongest biological oxidizing agent known because it has to instantly pull electrons from the most difficult source available, water.
Ah, and that's where the oxygen is released. The water molecule is split to fill that hole, releasing electrons, 2H+ ions into the thylakoid space, and the oxygen atom that eventually forms O2 gas.
The electron is now excited, and it falls down an electron transport chain from PS2 towards PSI. And you should think of this chain, which involves carriers like plastoquinone and the cytochrome complex, not as a simple path, but more like a hydroelectric dam.
I love that analogy. The energy released by the electron dropping is literally fueling a pump.
Exactly. The descent of that electron releases energy used to actively pump even more H+ ions from the stroma into the thylakoid space, which intensifies this proton gradient.
So you get this huge concentration of H+ inside the thylakoid, making it super acidic, so a low pH.
Right.
And that concentration difference, that potential energy, is what drives the ATP production through chemiosmosis.
That 1,000-fold difference in H+ concentration across the membrane powers the ATP synthase complex. The H+ ions rush back out into the stroma through this syntase, and that generates massive amounts of ATP. We call it photophosphorylation.
It's the same mechanism as in cellular respiration, just powered by light instead of food.
Identical mechanism. Now, the electron eventually lands at photosystem I, or PSI, which has the P700 chlorophyll. Gets re-excited by another photon of light and travels down a second, shorter electron transport chain.
Okay, so what happens at the end of that second chain?
The electron is eventually passed to an enzyme called NADP plus reductase. This enzyme uses the electron and two of those H+ ions floating around to reduce NADP plus to NADPH.
And this is crucial, right? The location.
So crucial. Both the ATP and the NADPH are produced on the outside of the thylakoid, in the stroma, which is exactly where the next stage of the process happens.
But there's a little footnote here, this thing called cyclic electron flow. If the linear path is so efficient, why does the plant sometimes use this alternative?
Well, cyclic flow uses only PSI, and it skips the water-splitting step completely. It cycles electrons back from a protein called ferredoxin to the cytochrome complex.
So it's a loop.
It's a loop.
Yeah.
And it generates extra ATP, but it produces no NADPH and no O2. The plant actually needs more ATP than NADPH to power the next stage, the Calvin cycle. So this is a crucial pathway to just balance the energy budget. It also offers some powerful photoprotection when light is too intense.
So we've successfully created our energy currency, the ATP and the NADPH. Now the second stage, the Calvin cycle, the synthesis part, occurs entirely in the stroma and uses that energy to actually build the sugar.
That's right. And this is an anabolic process. It consumes energy, it doesn't generate it.
And this cycle has to turn 3 times just to create a net output of 1 molecule of a 3-carbon sugar, glyceraldehyde 3-phosphate, or G3P.
And that's a key point. Many people assume glucose is the direct output, but it's not. G3P is the fundamental metabolic precursor that the plant uses for, well, for everything else.
Okay, let's track 3 molecules of CO2 through the 3 phases. Phase 1 is the initial incorporation of carbon. This is carbon fixation.
In carbon fixation, the 3 CO2 molecules attach to 3 molecules of a 5-carbon acceptor sugar called ribulose bisphosphate, or RoBP.
And this step is catalyzed by a very famous enzyme.
Probably the most famous, it's called rubisco-RoBP carboxylase oxygenase. And it's potentially the most abundant protein on the entire planet because it's so central to life.
Wow.
The resulting 6-carbon molecule is instantly unstable, and it splits into 6 molecules of 3-phosphoglycerate.
And that brings us to phase 2, reduction, where the light reactions currency is finally cashed in.
Right. The 6 molecules of 3-phosphoglycerate are converted into 6 molecules of G3P. This is an expensive step. It consumes 6 molecules of ATP and 6 molecules of NADPH to provide the reducing power and the energy to make the G3P.
And how many of those 6 G3P molecules actually get to exit the cycle to be used by the plant?
Only one. Just one of the 6 G3P molecules exits as the net carbohydrate product. The other 5 G3P molecules, which still contain 15 carbons between them, have to remain in the cycle to complete the third phase.
Which is regeneration.
Yes. The remaining 5 G3P molecules are rearranged to regenerate the 3 starting molecules of RuBP, the CO2 acceptor. And this rearrangement is itself metabolically complex and requires the input of 3 more ATP molecules.
Okay, so if we total up the cost for just one net G3P molecule, the plant spent 9 molecules of ATP and 6 molecules of NADPH.
It is a staggering investment. But the payoff is stable, storable energy for growth, for transport, and for structure.
Now, most plants, including major crops like rice and wheat, are known as C3 plants.
They're called this because the first stable product of carbon fixation we just talked about is that 3-carbon compound, 3-phosphoglycerate.
And the C3 pathway is fantastic under, say, cool, moist conditions. But plants in hot, arid environments face a huge trade-off, don't they? They have to close their microscopic leaf pores, the stomata, to stop losing precious water.
Closing the stomata is absolutely essential for survival, but it creates a crisis inside the leaf. The CO2 concentration plummets, and the O2 that's being released by the light reactions builds up dramatically.
And this leads to an evolutionary inefficiency known as photorespiration.
That's right.
Wait, if rubisco is the most abundant protein on Earth, why does it work so poorly in these conditions?
That's the evolutionary paradox. Rubisco evolved when the Earth's atmosphere had very little oxygen, so it was never really pressured to be strictly CO2 selective.
And it never had to be picky.
It never had to be picky. So when CO2 levels drop and O2 levels are high, rubisco mistakenly binds O2 instead of CO2 to the RuBP. This process consumes O2, uses ATP, and it releases CO2, yielding 0 sugar.
So it's an energy drain.
It's a huge energy drain. It can reduce photosynthetic output by half in some environments.
That sounds like a catastrophic design flaw. So how did plants in places like the tropics evolve solutions to get around this photorespiration problem?
They evolved 2 really brilliant strategies. First, you have the C4 plants like corn and sugarcane, and they use spatial separation. Their leaves have a specialized structure where CO2 fixation and the Calvin cycle happen in 2 different types of cells.
Okay, can you walk us through that spatial separation?
So the initial CO2 fixation happens in the outer mesophyll cells, but they don't use rubisco there. They use an enzyme called PEP carboxylase.
And that's the key difference.
It is the key. PEP carboxylase has an extremely high affinity for CO2 and absolutely 0 affinity for O2. So it successfully fixes CO2 into a 4-carbon compound, oxaloacetate.
So that 4-carbon compound is like an ingenious CO2 shuttle.
It is. The 4-carbon compound is transported into the adjacent bundle sheath cells, which are tightly sealed around the leaf veins. Once it's inside, CO2 is released at a high concentration, acting like a pump.
Ah, so you're flooding the zone.
You're flooding the zone with CO2. Because the bundle sheath cells are isolated, that high concentration ensures that when rubisco finally sees the carbon, it only binds CO2, which virtually eliminates photorespiration.
And I imagine that pump costs extra energy.
It does. The extra ATP required to run the CO2 pump is supplied by increased cyclic electron flow in these specific chloroplasts.
So that's a metabolic separation. Then we have cam plants, like cacti and pineapples, which use temporal separation, so separation by time.
Exactly. They conserve water by being active only at night. Their stomata open at night when the air is cooler, fixing CO2 into organic acids, which they then store in their cell vacuoles.
So they bank it.
They bank it overnight. During the day, Their stomata close up tight to prevent water loss, and the CO2 is released from those stored acids to supply the Calvin cycle. This ensures the cycle runs efficiently at the exact moment the light reactions are producing all the ATP and NADPH it needs.
What stands out to me when looking at the whole picture is just the stunning efficiency of this integrated system.
It really is.
The light reactions act almost like an instantaneous solar battery, fueling the slower, anabolic construction work of the Calvin cycle. And these ingenious C4 and CAM adaptations just rearrange the timing or the location to overcome these inherent evolutionary and environmental constraints.
Absolutely. And the sheer scale is hard to grasp. Photosynthesis generates an estimated 150 billion metric tons of organic matter annually. The sugars produced, that G3P building block, don't just create structural components like cellulose.
Which is the most abundant organic molecule on Earth.
Exactly. They also fuel cellular respiration for the entire food chain.
So, if we connect this fundamental biology to the challenges we face today, what does understanding the trade-offs between these different plant types mean for the big picture, especially with, say, climate change?
Well, this raises a really critical question. As global temperatures rise, they favor photorespiration, which is devastating for C3 plants like wheat and rice, our major food crops.
Right.
Researchers are now intensely focused on genetically engineering these key C3 crops to adopt the spatial separation mechanisms of the C4 pathway. So this suggests that understanding the precise, highly detailed metabolic steps we just covered might not just be textbook biology.
It might be the key to our future.
It might be the essential key to maintaining future global food security, something for you to chew on next time you look at a green leaf.